Lecture 1, Gene Prediction, Finding Genes and Open Reading Frames. One of the primary challenges in bioinformatics is identifying genes and open reading frames, ORFs, in a DNA sequence. 1. What is an ORF? An open reading frame is a stretch of nucleotides that begins with a start codon, usually August, and ends with a stop codon, UAA, UAG, or UGA. It has the potential to be translated into a protein. 2. Gene prediction tools. Glimmer, GeneMark, GenScan, Augustus. These tools use sequence characteristics like codon bias, promoter elements, and intron-exon structures to predict gene locations. 3. Applications. Annotating new genome sequences. Identifying potential coding sequences. Assisting in cloning and functional studies. Lecture 2, Microarrays and the Transcriptome. 1. What is a transcriptome? The transcriptome is the complete set of RNA transcripts produced by the genome under specific conditions or in specific tissues. 2. Microarray technology. A microarray is a collection of microscopic DNA spots attached to a solid surface. It allows researchers to measure the expression levels of thousands of genes at once. Fluorescently labeled cDNA is hybridized to the array. The intensity of each spot indicates expression level. 3. Applications. Comparing gene expression between healthy and diseased tissues. Studying cellular responses to environmental stimuli. Drug discovery and diagnostics. 4. Data analysis. Normalization, adjusts for technical variations. Clustering, groups genes with similar expression patterns. Heat maps, visualize gene activity across samples. Lecture 3, Applications and Future of Microarray Technologies. 1. Applications of microarrays. Cancer diagnostics, identify tumor-specific expression signatures. Pharmacogenomics, determine how patients respond to drugs. Nutrigenomics, study effects of nutrition on gene expression. 2. Future directions. Integration with next-generation sequencing, NGS. Miniaturized and portable microarray chips. Real-time expression monitoring. Use in precision medicine and epigenetics. Though NGS has taken center stage, microarrays remain a cost-effective tool for high-throughput gene expression studies. Lecture 4, Introduction to Genomics and Information Flow in Biology. 1. Genomics Overview. Genomics is the study of the structure, function, evolution, and mapping of genomes. Encompasses structural genomics, functional genomics, and comparative genomics. 2. Information flow in biology. The central dogma outlines the flow. DNA, RNA, protein. DNA stores genetic instructions. RNA transmits and interprets instructions. Proteins perform cellular functions. Computational biology helps to decode, analyze, and simulate this flow using large-scale data. 3. Experimental approaches. Whole genome sequencing. RNA-seq for transcriptome profiling. ChIP-seq for protein-DNA interaction mapping. These techniques generate vast data sets requiring sophisticated computational tools for analysis. Lecture 5, Genome Information Resources. To make genomic data accessible, several public databases and tools have been developed. 1. Major genome repositories. Ensemble, Genome Browser for vertebrates and model organisms. UCSC Genome Browser, visual tool for genome annotations. NCBI Genomes, genome projects from diverse species. 2. Data types stored. Raw sequence data. Annotated genes. Protein coding regions. SNPs and genetic variations. 3. Genome browsers. Allow users to search for genes. Visualize exon-intron structures. Examine sequence motifs. Access comparative genomics data. Lecture 6, Functional Proteomics and Protein Information Resources. Proteomics is the large-scale study of proteins, including their structures, functions, and interactions. 1. What is functional proteomics? It focuses on understanding how proteins function, where they are expressed, and how they interact with other cellular components. 2. Protein sequence and structural data. Primary sequence, linear amino acid chain. Secondary structure, alpha helices and beta sheets. Tertiary structure, 3D shape of a protein. Quaternary structure, multiple polypeptide chains in a complex. 3. Protein databases. UniProt, detailed protein sequences and functions. Interpro, functional domains and motifs. FAM, protein family classification. Protein Data Bank, PDB 3D structure coordinates. These resources are essential for drug discovery, molecular modeling, and understanding disease mechanisms. Lecture 7, Computational Genomics and Biological Data Analysis. Computational genomics applies computer algorithms to genome data to extract meaningful patterns. 1. Biological data analysis tasks. Gene prediction. Motif identification. Comparative genomics. SNP and variant analysis. 2. Internet tools and file formats. NCBI, EBI, and DDBJ offer online analysis tools. Common file formats. OFASTA, sequence data. OGFF/GTF, gene annotations. OSAM/BAM, sequence alignments. OVCF, genetic variations. Mastery of these file types is essential for bioinformatics workflows. Lecture 8, NCBI Model and Sequence Databases. 1. NCBI Structure. The National Center for Biotechnology Information, NCBI, integrates multiple databases through the NTRES search system. Key databases include GenBank, DNA sequences. RefSeq, curated reference genomes. GEO, gene expression data. PubMed, scientific literature. 2. Sequence databases. Store both nucleotide and protein data. Link to literature, taxonomy, functional info. Provide cross-referencing among datasets. 3. NCBI tools. BLAST, find homologous sequences. PrimerBLAST, primer design. ORF Finder, predict coding regions. These tools allow comprehensive biological investigations from any web browser. Lecture 9, Sequence Alignment Tools, FASTA and BLAST. 1. FASTA algorithm. Introduced by Lipman and Pearson. Efficient for identifying similarities between sequences. Uses k-tuples, short words, to initiate alignments. 2. BLAST, basic local alignment search tool. Compares input sequence against database. Fast and widely used. Returns list of similar sequences with evalues and scores. Types of BLAST. BLASTn, DNA versus DNA. BLAST, protein versus protein. BLAST, DNA translated versus protein. BLASTn, protein versus translated DNA. BLAST, translated DNA versus translated DNA. 3. Multiple sequence alignment. Tools, Cluster Omega, MOSO, MAFT. Applications. Phylogenetic studies. Domain conservation. Mutation analysis. Lecture 10, Structural databases and protein modeling. 1. Small molecule databases. PubChem, chemical structures and bioactivity data. ChEMBL, drug-like bioactive compounds. Zinc, ready-to-dock compounds for virtual screening. 2. Protein Data Bank, PDB. Stores 3D coordinates of biomolecules. Formats, PDB, MMCIF. Supports tools like PYMOL, camera for visualization. 3. Protein modeling. If structure is unknown. Homology modeling, based on similar known structure. Ab initio modeling, predicts from scratch. Tools: Swiss model, Fire2, ITASA. Protein structures reveal. Active sites. Binding pockets. Allosteric regions. These insights guide drug design and synthetic biology. White heavy checkmark Module 2 Summary. In this second module, we explored 1. Gene prediction and the concept of open reading frames. 2. The role of microarrays and transcriptome analysis. 3. Introduction to genomics and biological information flow. 4. Resources for genome and protein data. 5. The domain of functional proteomics. 6. Tools and file formats used in computational genomics. 7. Applications of FASTA and BLAST for sequence comparison. 8. Structural databases and protein modeling techniques. Graduation Cap Final Course Wrap-Up, Computation Biology and Bioinformation. Throughout both modules, we've built a strong foundation in molecular biology concepts and genome organization, DNA and protein databases, sequence alignment, gene prediction, and phylogenetics, functional genomics and proteomics, bioinformatics tools and file formats, applications in drug discovery, diagnostics, and personalized medicine. You are now well prepared to use computational tools for solving real-world biological problems.